19, at the end of it, it was alleged that there's a new kind of disease-causing microbe particle on Earth. There are always cases of pneumonia in humans where no known cause, no known microbe can be associated in any detection process or the general detection process, the PCR detection. There are cases of these in China, in Australia, everywhere, where someone has pneumonia, the clinical symptoms, and they don't pass positive on the test for any of the known causes. And this is unknown pneumonia. A story in China developed enough energy to the point where a team got together and grabbed 3 of these cases, or 4 actually, but only 3 were used, of unknown pneumonia, where people had pneumonia without a diagnosis. The specific team made sure that the 3 cases had a connection to the wet market, the seafood market in Wuhan, and then that provides the hypothesis that there's a new microbe out there that is causing disease, because there is a lot of disease out there, especially respiratory disease, and people complaining, the public complaining, saying, please fix this. So a new disease particle is profiled, enough energy is put together to have this take place in Wuhan. These 3 people are officially collected, and their respiratory sample is mixed together and coagulated, centrifuged, so that the RNA particles of the sample can be concentrated. And these particles, that they are, along with some antibiotics to make an antifungal kind of materials to make sure whatever does grow, because the respiratory sample is not going to be perfectly clean. So any fungus or bacteria in there, we have to put materials in the collection when we try to propagate the alleged new virus. We want to include these other materials like antibiotics so that only the virus can propagate on the cells. The cells that were used in the Wuhan investigation, when they come up with this new signature of a microbe, those cells were lung cancer cells of the airway, of the outside cells of the airway, taken from a patient who had lung cancer in surgery. These cells are made into a lawn, passage 1 cells, which means it's something like they've only just divided, or they're ready to divide again, I'm not sure. But they were cancer cells, and the centrifuged samples from the 3 people who had pneumonia, with the idea that there's a new microbe in them, and that it started in the wet market in Wuhan, and that's why these 3 people are sick, because they have a connection to the wet market. Their samples are put onto the cells, the cell lawns, and a process takes place. They're incubated at 37 degrees Celsius. They're washed to get any unbound virus off, and then they're left for some time. Actually, I've emailed the authors of the study. I don't think they've made it. They haven't actually said how long the cells were there for. I don't think. I can show you. They were incubated for 2 hours, and then the virus was washed off them. And then for some unstipulated or undisclosed amount of time, every 48 hours, the cells were washed with saline or fed, I don't know how you'd call it. And then eventually, after 10, anyway, I emailed them about this because I think it's not written correctly. The details aren't all there. But anyway, you get the basic picture. The cells have the material put on them and then washed, and then they're left for a while, and then we come back to see that the cells are actually, they look different. There's an effect on the cells that we have put the material on, and that's just about that. So from here, think the particles that are resulted from this cancer cells that had the samples put on them, they're looked at, and not one of these particles is actually taken and analyzed in itself. All of the biochemical analysis that takes place is analyzing the collection of material, and pieces are collected from the collection and put together as a model to make a genome. Some of them showed cytopathic effects. So I'm saying when we put the sample on these cancer cells, the cancer cells did break down to an extent where the material touched them. And this would be expected even if the viruses weren't all that the mainstream teachers are saying that they are, because the cells are chemically fragile. They've had some stuff added to them and been put through this process. The ones that did have stuff affect them or touch them, proceed through them, they ended up crimped, a bit damaged compared to the ones that didn't end up touching the stuff. From here, this viral genome sequencing takes place from all these particles that are harvested from the cancer cells that had the patient's sample put on them, centrifuge sample supernatant. All of these particles that you can see in the micrographs, in the graphs, here's these particles in the slides. They put dye with them and they take photos of them. Like I was saying, not one of these particles is taken and thoroughly analyzed. It's the collection of them. But this viral genome sequencing, I don't know how this stuff works. The doctor, Stefan Lanka, seems to know what it's all about, and he reckons it's all just, I think he was saying, unless he was talking about something else, that there's a lot of imagining what you've got, what sample it is, and finding all the readings that come from it and putting those readings together as if they fit into what you were imagining was actually already the case or going to be the case. But I don't know how it all works out. It could actually be that the cancer cells that break down are slightly different. They come from a different patient or from a different line, or they're treated slightly differently, and it results in just a slightly different soup, slightly different readings, although very similar, because look at this. These are all the readings from previously. So all this in orange here is the new, that's the new coronavirus, and this is the original SARS, and these are all other things, other coronaviruses. This is a genetic map. So here is the relation between SARS-1 and SARS-2 is evident, allegedly here. The difference, the distance. I'm not familiar with reading these maps, but It could be that every time you take a reading of the same thing, you're going to have slight differences too. I'm not sure. This, I must humbly say, is not my field. I can't really comment much on this. It's just, if it all comes down to this though, I'm not convinced because I don't even understand it. And even if there are, even if it is a new kind of particle, it doesn't mean it's causing disease. The big deciding factors of whether I'm going to believe in a disease-causing particle comes down not to genomic reading of particles, but rather the effects that these particles have pronounced in controlled experiments, which I've spoken about on my YouTube. That's basically it. The coronavirus itself wasn't taken from a human scene. The alleged new coronavirus was allegedly taken from 3 humans who were at the same market, and it was allegedly put onto some cancer cells. The cancer cells were allegedly infected and mass propagated this virus, and these are all these kinds of particles. And not one of the particles can be read and biochemically analyzed in itself, but measurement of all the particles altogether, readings from that, we've put together a signature, and apparently there's a new disease-causing virus out there. Apparently it has bristled the fur of some mice, some genetically modified mice. Yeah. It came from a bat, and then it's infecting humans, and then apparently the humans are seeing if it's a legitimate infective agent by putting it on a mouse. This is just bizarre. From day dot, we haven't even reenacted a virus disease being transferred by adding a virus. And you might say, how do you do that? Well, you create 2 conditions exactly the same, except in one of the conditions you are introducing the virus, not in the vein, just at a quantity, at a kind of rate that occurs only in nature, and in a fashion that occurs only in nature, not injecting it inside its vein, not dripping it down its nose while its head is locked in one position. I'm talking about putting it in the air. I'm talking about getting a fish tank and 2 that are exactly the same, other than one has introduced a virus in it. Virus, bacteria, fungus, any of it. This whole virus delusion has come about because we've seen funguses and bacteria. They are real things. They're big enough to notice that they're real. We can individually analyze these things. We sprinkle them onto host organisms and nothing happens. We jam them on the host and disease is caused, and some people say, yeah, that's how it works. In fact, not many people really know about these experiments, but the great majority of veterinarians, vet clinicians will tell you, and doctors will say, yeah, it's happened like that. It just happens like that with pronounced disease, but they don't know the nitty-gritty details of the absolute absurdity. We come along, sprinkle bacteria on humans, and nothing happens. Yet we've decided that the bacteria is the cause of disease, because what else would it be? For thousands of years, we've been blaming external entities. Here we've now found the microbes that are unequivocally associated with disease. The disease that comes without a trace. That's what I'm talking about. That suddenly you're sick, you don't know why. Well, it's come from somewhere, we've thought, for thousands of years. Recently we've found the microbes that are only there when the disease is there. And we've blamed them and we've sprinkled them on people and controlled experiments and nothing happens. Same. We have these viral particles that we've nowadays can see. And we've sprinkled them on humans and nothing has happened. Same deal. So we've jammed them on humans. We've jammed them inside veins and the host dies. Same dynamic as bacteria and fungus. But they're so small, this seems to have evaded us that they're actually not infecting cells and being mass reproduced, it seems. How is that possible that's even possible? If the whole majority of the whole world's saying this is how it works, viruses infect things, how is it possible that maybe they don't? Because this, as I'm showing you, this is all we know. This is the closest we can get. We can't see them infecting cells. We can't even biochemically analyze one of them itself. And then how, if they're not really infecting cells, if they're just cellular debris, how have we come up with this big theory? Because we already thought bacteria and fungus were causing all this disease. And they're just particles that we can see, and these are too small for us to be able to see that they're not even doing what we think they are, if they really aren't. They are that small, it's evaded us. And the idea that they've caused the disease that comes without a trace has evaded us just as well as it did with bacteria and fungus. Except here the particles are that small. When we finally collect them, they do the same thing as bacteria and fungus. This virology is the flowering of the germ theory of delusion, the germ theory of disease, I mean. The virology is the flowering of that. The particles, we knew that they were there from tobacco mosaic virus. There was something there that we couldn't see that was killing the things. And now we realize they're too small for us to know exactly the ins and out. But as I'm saying, this is as close as we can get, and it looks pretty well unequivocal that, I mean, when you include the challenges, the reenactments of where allegedly these viral diseases take place in nature, when we reenact those scenarios, and when you see actually what we really do know and what is actually all just bluff, you might end up like me and think, oh yeah, I think that's how it works. If you do, yeah, hopefully you don't just jump there, as a lot of people are. Yeah, I realise how it works, everyone's lying. There's very intimate details here. This is not a big conspiracy theory as far as I can see.